right. Hello, everyone. I'm glad we had a little movement break here, and I'm really glad that everyone was able to stick it out here to the end of the day. I know it can be really rough trying to stick through all these different talks, but we've had some really amazing speakers here today, and so I'm really glad I get to be sharing the stage with everyone. So today I want to talk about gene therapy, what it is, the technology that's been developed to help us get to where we are today, and how we're using this approach for Parkinson's disease specifically. And just a little bit of background on me. So I'm a movement disorder specialist, and I've been having training, working with a lot of patients like you in the clinic. And there was a point where I was starting to get a little frustrated working with patients, just handing out more Cinemet, trying to treat their symptoms. And I'd like to think that we did do a decent job getting people to have a better day. But overall, it was starting to get a little bit frustrating, not being able to actually alter the disease course and not able to really address the underlying causes of Parkinson's. And that's what really led me to be interested in pulling out what I was doing in the lab and with my research and bringing that into the clinic so that we could really try to make some headway on affecting progression of Parkinson's. And this is really what's bringing me and talking to you here today. So these are some of my disclaimers. I'm an employee of Asklepios. I think you may have seen some of the icons that we have available here today. And we do not have a product, so I'm not promoting anything. I just want to give you a brief update on what is Parkinson, or what is gene therapy and how we're using it for Parkinson's. These are more disclosures. Happy to discuss them if you're interested. So I just want to take a quick minute to talk about who is Asklepios. I think many of you probably have never heard of them before. I want to talk about gene therapy and why we'd even think about it for Parkinson's disease. And then really, how do you do gene therapy? It's not so easy. And at the end, I want to wrap up with how you could get involved with other clinical studies. So just first, a quick minute on Asklepios. I mean, I'm guessing almost no one here has probably heard of Asklepios. This company's actually been around for about 20 years and really focused on gene therapy, but only recently getting involved with Parkinson's, and this is where I'm coming in and speaking to you today. But Asklepios was the first to clone AAV. This is the packaging that we use for gene therapy, and I'll be talking about that a bit more. But the first to deliver it in some unique ways, so into the spinal fluid, but also to the brain directly, one of the first groups to do that ever. And able to do this because we have a really robust toolbox. We have lots of tools that we can use to make the best type of gene therapy for a variety of diseases. And so that's allowed us to have this really diverse pipeline. And so right now in clinic, we have gene therapies for multiple types of diseases. So everything from muscle diseases to heart failure to other brain diseases, including Parkinson's that we'll be focusing on today. And one other unique thing with Asklepios is that they're able to manufacture the gene therapy internally. So this is an end-to-end company, which is fairly unique in this world. Just a couple other things. So there's actually 4 different countries where Asklepios is located, in Spain and France and Edinburgh, Scotland, but also headquartered in North Carolina, and really pioneered by Jude Samolsky, who discovered AAV. And using AAV that has been now used in all the gene therapies that have been approved. This technology was really built off of this platform we'll be talking about today. And this is really what has made Asklepios a leader in gene therapy. And if you spend any time looking at Asklepios, you'll quickly realize that Asklepios was actually recently acquired by Bayer. And this has actually been a really great move because it provides the resources needed to develop these very complicated gene therapies that you'll be seeing here in a minute. So before I can tell you what gene therapy is, I need to tell you what it isn't. There's a lot of, I think, misconceptions and myths out there as to what exactly is gene therapy. So I'm not talking today about gene editing. This isn't CRISPR, for example. This is not cloning. We're not making Dolly the sheep. This is not how you make designer babies, though that would be interesting. And I'm not talking about stem cells. So if we're not talking about all these different things, so let's put all those aside, what are we talking about when we say gene therapy? So come back to class with me for a minute. So what are genes? We have to make sure we're all starting from the same page. So what are genes? Genes are DNA, and this is our instruction book for life. This is how every cell in all of our bodies know what to do. These instructions are used for the normal growth and health of all of our cells. So think of genes as our DNA, and our DNA is made up of lots of different genes, and this is the blueprint for every cell in our body. So when we have genes, the cells just naturally, their natural function here is to read the genes. They read the instructions, and from that, they make proteins. And proteins function as the business end of a gene. Now, there are lots of kinds of proteins out there. And proteins do everything from signaling the cell, the health of the cell, to every other function that's needed to make a cell work. And it depends on what kind of cell it is as to what kind of function we're asking it to do. And gene therapy takes advantage of this natural occurring process in every single cell and uses this ability to read genes to basically take that gene that you want to put in that cell and then effectively use that as your way of giving a new kind of medication. So again, genes are our instructions. They're important and required for the health and growth of all of our cells. Now, how we use gene therapy to our advantage, and we can use this in a variety of diseases in many different ways. So if you had a gene that was mutated or needed to be corrected, wasn't working in some way, you can effectively restore function this way. If you had a gene that's behaving badly and you want to quiet it or inhibit it, you may want to inactivate the function of this bad gene. In other cases, you may want to add a gene in to actually enhance a function, or you modify the function of a gene. And today I'm going to be talking about two different gene therapy studies for Parkinson's, one that's looking at correcting an abnormal gene, another that's enhancing function of a gene. So how exactly do you deliver genes to brain cells? And I'm going to tell you this has taken decades for us to figure out how to do this and do it well and effectively and repeatedly. So what is gene therapy and how are we doing this for the brain? So with gene therapy, we're taking a healthy new copy of DNA or gene as a new way to treat disease, so using genes as medicine. We're taking this healthy gene and we have to package it up, and what we're using is an emptied viral vector. So we're taking this healthy gene and using the vector or this packaging to help get it into the cells that are being affected by the disease, in our case, Parkinson's. What we're using for our packaging here is called AAV, or adeno-associated virus. This is used by Asklepios, but many other gene therapies have used this type of delivery system. AAV has been actually great and has been revolutionary in really bringing gene therapy more to the forefront, mostly because it's shelled out, it's empty. We're just using the outside of the packaging, and AAV by itself cannot cause infection in humans. So we have here on the right this big red and purple. This is what an AAV looks like under a really good microscope. I want you to think of it like it's an envelope or a packaging. So AAV is, again, like an envelope, and we use AAV2 specifically often for the brain. And we do this for a variety of reasons. So one, AAV is really good at helping genes get into brain cells. Not every packaging is able to do this for brain cells specifically. And it took us a while to figure this out, but it's really good for if we want to be studying brain diseases like Parkinson's. It's been safely used in many other neurologic gene therapy studies. So we have some pretty decent experience using this particular packaging for a variety of brain diseases. And the nice thing about working in the brain is that brain cells are not turning over. They're not dying and coming back like blood cells or something like this. And so the hope here is that it's a very lasting and durable effect from our gene therapy. So I'm trying to think about this in a slightly different way. So we have the purple ladder-looking thing. That's our DNA or our gene. This is like a letter, and we want to package it into our envelope. And so we can do this, as you can see here, put our letter together. And now we need to address it and get it to the right location. And in our case, and what we talk about today, is we actually use surgery to help deliver our gene therapy directly to the part of the brain that's being affected in Parkinson's. And then we ask our brain cells to be the readers and then to read the gene of our interest, again, using genes as medicine. So looking again, we got our little piece of DNA. This is our gene of interest. And this is our package, or the AAV. Now, these two things actually really like to come together, so we can put this together essentially in a test tube, if you will. And then we use surgery to get it to the cells that we want to be taking up the genes of interest. Once there, we know that viruses are really good at getting genes into our cells. That's how they make us sick. But in this case, we have a shelled out empty vector. We're just using it to deliver the gene that we want. And once it's in there, all the packaging gets taken up and used up, and all we're left with is just the DNA. And we know the cells know what to do with this because they read DNA all day long. And in this case, we're asking it to just read the one gene that we made, and this is, in this case, GDNF, which we'll be talking about in a minute. And it reads this, and it makes the protein that we want, and in abundance. So why would we even think about gene therapy for Parkinson's? You know, I think there's a lot of people who feel that gene therapy is only for genetically based diseases. And though we know that there are a lot of genes involved with Parkinson's, it may not be like the only cause for the vast majority of patients with Parkinson's. So I think it's quickly put away. I think there's a lot of reasons we could use gene therapy to treat a lot of diseases like Parkinson's that don't have a pure genetic basis to it. Now, one benefit is, again, this is a one-time treatment that we expect to last a very long time. Again, the brain cells aren't turning over. They're not dying and coming back. And so the gene that we put in there will stay there. And in fact, actually, and many of you may not know about this, but gene therapy has been used in Parkinson's since 2003. So everything I'm describing here may seem very novel, but in fact, we've been working on this for quite some time. So in the past, what Parkinson's gene therapy has really been focused on is two different ways of trying to help Parkinson's disease symptoms. So one has really been used to increasing levels of dopamine in the system, so effectively working like Cinemet, let's say, trying to boost levels of dopamine back up to help with symptoms, but this primarily only works on the motor symptoms like tremor and stiffness and slowness. And then there are other gene therapies, then some of which I'll be talking about today, that have been really focused on ways to slow down disease progression of Parkinson's. And so here we are in March of 2022, and there are two gene therapy studies that are actively recruiting for Parkinson's disease specifically. So one is looking at AAV for GBA, and this is really focused on patients that have Parkinson's and a known GBA1 mutation. I have a few more slides coming up to describe this a bit more. But this is effectively, like I showed you that first line, where it's trying to correct a mutation that's there. So we know there's a mutation. We're trying to overcome it with gene therapy. And the one that's being sponsored by Asklepios, which I will be talking about a little bit more here today, is AAV with GDNF. And this is for neurorestoration, trying to restore the dopaminergic system. I just wanted to note, because I think this is interesting, that a lot of other gene therapy studies in the past have been focused on trying to boost dopamine levels, but none of them are active at the moment. Just wanted to point that out. So first I want to talk about this AAV9 with GBA. So this is sponsored by Prevail Therapeutics. This is still very early stage. This is a phase 1/2A, and it's recruiting in the United States and also in Israel. And they're looking for patients that have a known GBA1 mutation. And so, though I think that this is really important for patients with GBA mutations, not everybody either knows that they have it or will have a GBA mutation. So not everyone will be able to qualify for this. But one unique thing is that they're putting it directly into the spinal fluid. And again, the idea behind this is to try to get the gene therapy to go over the entire brain, because we know it's not just the dopamine cells that are affected in Parkinson's disease, and this is one way to potentially address that. And so using in Parkinson's disease with a known GBA mutation, this is a situation where patients aren't making enough of something called GCase. And without GCase, we don't have a very good garbage disposal system working in those neurons. And without a good garbage disposal system, that means that things like alpha-synuclein, which you've heard quite a bit about today, that gets junked up and clumped up inside the cells. And so the idea behind GBA gene therapy is that we are able to really boost the levels of GBA. That allows GCase levels to go up. And that is the thinking behind this, and the literature would say that this is going to help the garbage disposal system work better. And with a better garbage disposal system in the dopamine neurons, this is going to help potentially slow down progression of the disease. And we'll do this by hopefully reducing inflammation in the brain, which we just heard a really great talk about, but also potentially slow down the accumulation of alpha-synuclein inside these cells. And again, the primary function here is we're trying to slow down the progression of disease. So that's the GBA, gene therapy. So the gene therapy product that we're working with and that SBio is sponsoring is AAV2 GDNF. This is also a very early stage, so this is a phase 1B. There was an earlier study that was done at the NIH that's just now wrapping up. So this is an extension of that study, now looking at earlier stages of Parkinson's. We're nearing completion of the study, and we're only recruiting in the United States. So we're looking for earlier stage patients. Again, this is a growth factor, and so the idea here is that when you are earlier stages of disease, that there's more to work on, there's more to respond to a growth factor. So we're looking for patients with PD with more mild to moderate types of symptoms. So what is GDNF? So we actually found out back in the 90s that GDNF is essential. It's required for the development of dopamine neurons. And this is in spite of the levels that are needed for the development of the neurons. We know that GDNF is also just needed even in the adult state. And so levels of GDNF are actually really high when you're young, when the brain is growing and still developing and all the wires are being connected. But even as adults, we still need a little bit to maintain the health of our dopamine neurons. And it's the hope here that with more GDNF, and we've seen this in many animal models in the lab, that GDNF and like growth factor will help alter the course of Parkinson's and not just treat the symptoms. And so for our GDNF program here for Parkinson's, the thinking here is that there is a loss of GDNF, and there are many reasons why Parkinson's happens, but one of them may be that you're losing the growth factor. There's just not enough of the food that's needed to help the cells remain alive and healthy. And so with this investigational gene therapy, we're hoping that the cells that are there, that are still healthy and they're kind of sticking in, that they can respond to the GDNF and become healthier as well. The idea with gene therapy for GDNF is that we're able to really boost the levels back up to where you were when you were young. So that higher levels of GDNF have then the potential to restore these sick but not yet dead cells that are remaining in the Parkinson's brain. And again, this is where we hope that it opens the possibility that we can actually start altering the course of Parkinson's and not just treat the symptoms. And so this is a question my mom posed to me. So, okay, that's all great, but how do you do gene therapy? And how do you do it in the brain? And so this is what I'm showing you here has taken 30 years to get to this place. And we're actually doing gene therapy delivery right into the part of the brain that's being affected in Parkinson's and doing it inside an MRI machine. So this is all done with MRI monitoring. And so patients who go through a really rigorous screening process to make sure that you would be safe and that this would be a good study for you to be in. And if a participant qualifies, they're invited to come into the study. And this is a one-time administration of gene therapy directly to the part of the brain called the putamen. The surgery happens in the MRI machine while you're asleep. So using this MRI imaging, we can see exactly where we're going and what we're doing. This is a huge advantage because it allows us to customize each infusion to every putamen for every individual. And I think one other thing to point out, this may still seem sci-fi to me sometimes, but this has been safely used for over a decade. And not just Parkinson's, but a lot of other brain diseases and other things like brain tumors, for example. And so what we're trying to do in these surgeries is we're trying to get to a very precise part of the brain and deliver the drug right and only in that one spot in the brain. And so what I've circled here in red is the area called the putamen, and this is where we're trying to get our drug and not trying to get it everywhere. We're trying to get it just at this one spot. So a very precise delivery of our drug to the brain. So what does this look like? And this is I'm trying to highlight the slightly darker gray blob here. So those are the putamen. You have one on each side. And what we're trying to do is fill up that putamen with white. And you'll see this in a video here coming up. What we're trying to do is just one infusion for each putamen. And what we're doing here is what we call shape fitting. So we're trying to get the drug only into the putamen. You can go ahead and play the video. And so you can see the white coming in. That's actually a signal to us of where the drug is going. And what we're trying to do is just fill up that putamen, or that area that I had circled in red. And so what you're also seeing here is, again, decades of development of technology and all the tools that are needed to do this safely and repeatedly. And so what we're seeing here are two hollow tubes that are being inserted directly into the putamen. And because we're doing it in the MRI machine, we can see exactly where those tubes are and where the drug is going. And so if we can sometimes make changes or adjust the infusion or how fast we're delivering it in order to be able to make sure we're keeping it inside that person's putamen. Now, this video is playing pretty fast here, and so I just want to point out this actually takes place over a couple of hours, so it's really sped up. But I think, what's been impressive to me and what has been impressive to other patients that have seen this is that we're able to really customize this for every individual. Now, the other big change that has happened from other gene therapy studies is that we're actually delivering this in the back of the head, so we're going from the behind. And this has actually allowed us to do just one pass of our cannula, or this hollow tube, to each putamen. And that means that we're able to do this a little bit faster. And it's also allowing us to get more of our drug into the right place. We're able to get more than half of the putamen covered this way. And we think this is really important for Parkinson's in particular, to really make sure we're getting the best benefit that we can for Parkinson's patients undergoing this procedure. So why are we taking so much care and so much time, and why does this really customized delivery matter for GDNF? Well, it's, again, decades of development of this technology to help us get here. And so we really look at the convergence of all these different technologies that have helped us finally reach this place. And so, we do think that we found the right drug. We think GDNF may be the right thing for Parkinson's disease. Again, we know that this is essential for the well-being of dopamine neurons. And now with use of AAV, again, this has been used for many years in the clinic as well, and that we have the right packaging and the right vehicle, the delivery material, to get our gene into the cells that we want. And now we also have developed the right type of delivery. Again, this has taken decades of getting us to this point, but now we have the ability to do this really precise injection into very precise parts of the brain. And with this, we now have the better delivery with the right drug, and now we have the right way to dose this. Because getting the right dose of gene therapy isn't so straightforward, especially when we're delivering it to part of the brain, and we think that we finally have gotten to that place. And with that, we think that we now have the ability to potentially really address the underlying Parkinson's disease, and we really hope that this will mean better outcomes for patients with PD. Now, we've had this study running for a little over a year now, and just about to wrap up enrollment. I really hope that I can come back next time and I can show you some of the results of what we've been getting, and we'll be showing some of this data in some upcoming meetings for the Mood Disorder Society. I do want to take 1 second, though, and talk about a pre-gene therapy natural history study. This is kind of a unique thing, and I want to introduce this since we have a site we're opening here in Florida. So we're looking for patients that have both Parkinson's or MSA. So a lot of these, again, we're looking for early stage disease because of how we think this growth factor will work. And in very early stages, both Parkinson's and MSA can look very similar. They're seen by the same movement specialist. There's lots of reasons why there's a lot of benefit potentially to look at both of these diseases. And we're looking to enroll these patients into this pre-gene therapy study with the idea of being able to capture a lot of information about how these diseases are changing over time before we then enroll them into a GDNF gene therapy study. So right now we're looking for patients, in this case with Parkinson's, for patients that are in the early kind of middle stages of Parkinson's, so 1 to 6 years from diagnosis. And we're looking for people who are already on levodopa. So this isn't for people who were just diagnosed or just kind of figuring things out. This is for people who are in it a little bit and kind of understand the ebb and flow of the day-to-day. And what we're trying to do is really monitor changes in clinical assessments, a lot of things that you may be doing with your neurologist now, a lot of motor exams, but other just questionnaires and looking at MRIs, and then also looking at biomarkers in both blood and spinal fluid. And again, the idea is if we're really going to be altering the progression of disease, I need to know where things were before you got treated. And this is our opportunity to try and capture some of that information so that we have a better sense of what our drug is doing down the road. There's a flyer that should have been put in your packets about this as well, and I would encourage everyone, if you're just interested, please feel free to reach out to AskBio. And so I've talked about some neurosurgical gene therapies here. This may not be everybody's cup of tea, I get that. But even if that's not it, I would encourage everybody here to think about joining a clinical trial. There's no way we're going to get better about getting ahead of Parkinson's disease if we aren't actually able to do the research. A lot of these studies get slowed down or do not finish at all because people aren't able to participate. They can't recruit well enough. So please just think about it. If it's not these, that's fine, but something. Even if you're a healthy volunteer, it still helps. And so this was a really stunning statistic for me, so I have to share this everywhere I go. But if I were to ask this room, everyone, about 60% of people with Parkinson's would say, yeah, I'll join a clinical trial. I'd think about that. Yet at the end of the day, it's only about 10% of patients with Parkinson's that actually sign up for a clinical study. And so I think that was real jarring for me, so hopefully this will encourage some people to consider joining a clinical study. Now, I would encourage everybody to try and do their own homework, and if you want to learn more, this is a great place to start. So this is clinicaltrials.gov. Every legitimate clinical study is required to be signed up here at clinicaltrials.gov. So if you're thinking about something and you want to make sure it's legitimate, you should check here at clinicaltrials.gov. Now, this is a great website. There is a lot of information. However, it can be a little bit hard to maneuver. And so I would also recommend looking at the Fox Trial Finder. So this is actually a bit more user-friendly. They have great ways of mapping out where studies are. So you can say, I'm located here in Florida, what do you have near me? And you can filter out and pick out different types of studies that might be a better fit for you. They also have studies for MSA and ways to get involved if you're a healthy control, if you're a spouse of a patient with PD. So please go there and check it out. And if you have any other interest, want to learn any more, please just tell your neurologist you're interested. I think sometimes there's not enough time in clinic to discuss this. But if you tell your neurologist that you're interested, this is a great way to start that conversation, and they can at least help direct you to the right place if desired. I'm happy to talk to anyone after we get done talking here today if you're willing to stick around a little bit more. And otherwise, feel free to email us at this askfirst@askbio.com. This is also listed on the flyer that's in your packets that you received today. We have a newsletter so you can just keep up to date with some of the events that we have going on right now. But if you have questions, this is a great place to answer. Matt is the one who answers this email, so we'd be happy to answer any questions you might have. And with that, I'd say thank you for sticking it out this late afternoon on such a beautiful day. It's been a real pleasure speaking with you.